next non-Mendelian inheritance is pleiotropy. Pleiotropy nothing but the single gene product may produces more than one phenotypic effect. So if the single gene product it produces more than one phenotypic effect in organism, that phenomenon we can call it as pleiotropy. So the genes, the gene which is responsible for multiple phenotypic effect, we can call it as pleiotropy gene. So here, in most cases, the mechanism of pleiotropy is the effect of gene on metabolic pathway and which contribute towards different phenotypes. For this, we will see some example. Starch synthesis in pea plant. It's a very good example for pleiotropic gene. And another examples are phenylketonuria. Phenylketonuria. And another example is sickle cell anemia. Sickle cell anemia also example for pleiotropic effect. So this phenylketonuria and sickle cell anemia are genetic diseases. So in this particular disease, the mutant gene exhibit the it's a many phenotypic effect. For example, if you take phenylketonuria, so it result in mental retardation, a reduction in hair, and a reduction in the skin pigmentation. So these are the different phenotypic effect produced by single gene. So single gene product. That's why this phenomenon we are calling as pleiotropy. Okay. So here we will discuss the starch synthesis in pea plant. Actually, starch synthesis in pea plant is controlled by two alleles. So the two alleles, one is a BB. So caps B, the gene is BB. Here the dominant alleles are, it's a caps B, and the recessive alleles are small b. So this gene, the product on the which is responsible for the synthesis of starch in pea plant. So it depends on the dominant recessive allele, the amount of starch may be produced in the seed of the pea plant, right? Pea seeds, right? So if the pea plant with the dominant alleles BB, which produces large size seed, and the seeds also look like round shape. The shape of seeds also same round. Why the seeds are large? This gene, the BB gene, they efficiently produces the starch in the pea plant. Because of high efficiency, because of more synthesis of starch, the seed look like large and also looking like brown color. But the pea plant with small p genotype, this plant produces seeds which are very small and also it's a wrinkled shape. So wrinkled shape seeds. Why the seeds are very small? So these are recessive allele. So it's synthesis starch very less efficient. Starch synthesis in this plant is less efficient, so that the seeds look like very small size and also wrinkled one. So when we are making cross in this particular gene, okay, see if these are pure breeding pea plant, these are also pure breeding pea plant. This is a parent organism, right? Pure breeding. So this plant, particular plant, it produces gametes, only one kind of gamete, because these are homozygous dominant, these also homozygous dominant. This one produces gamete with recessive B allele, right? These are the gametes. When we are making cross between this large and round seed of pea with this small and wrinkled seed, we will get F1 progeny, the heterozygous progeny we will get. So the heterozygous condition. But these particular seeds, how it look now? They look so intermediate and also round also. Intermediate and also round also. But here you can see the same gene pair only. So the caps B and small b. But these caps B dominant when present in homozygous condition, which producing large size seeds. The large size is a phenotype. But here the size is somewhat reduced. It's not looking like small, not looking like large. So that it producing another one phenotype So because of that phenotypic change, the size of seeds on the looking like intermediate and it produces round also. Okay, we will see in F2 generation how it will produce us. This F1 hybrid produces two types of gametes, caps B and small b. We put nuts here. Here you can see caps B, small b, caps B, small b. Caps B, caps B, caps B, small b, caps B, small b, small b, small b. Here the caps B is it's a large and round seeds, and these caps B and small b is seeds are its intermediate size and round, and small b here is they are small size and wrinkled seed. So that the phenotypic effect, so the phenotypic ratio what we are getting is 1:2:1, and the genotypic ratio also, so 1:2:1, right? So this phenotypic ratio, like it's deviated from the Mendel, because as per Mendel law of dominance and segregation, as per monohybrid class, you have to get only two types of phenotypes only, either a dominant phenotype or recessive phenotype. But here we are getting additionally another one phenotype, like dominant. Here one is dominant, large and round, and this one is a recessive phenotype, small and wrinkled. But this particular gene, if they are present in heterozygous condition, it producing another phenotypic effect. That's why this gene we are calling as pleiotrophic gene. The phenomenon you can call it as pleiotrophy. I hope that you understood the topic, right? So next we are going to discuss about concept of dominance, right? And we know that in organism, the particular trait is either controlled by or expressed by pair of dominant alleles or pair of recessive alleles. But dominance is not a autonomous feature of gene or product. But every gene in the given organism contain information to express particular trait. Like you see, each and every character or trait of organism is expressed only by the pair of genes only. If you take any diploid organism, they have two copies of genes for particular trait. The two copies of genes we are calling as alleles. For example, the pea plant, the gene responsible for tall height is caps T. So this pea plant is diploid organism. So the diploid organism, they are having two copies of gene for particular trait, nothing but plant height. So these particular copies we are calling as alleles. We are saying that these alleles either may be identical or may be non-identical. So the alleles you can see for height either may be caps T or small t. In these cases, these alleles are identical allele. Here both are dominant allele, here both are recessive allele. Or the alleles may be in that pair, maybe it's a non-identical also. Caps T and small t is there. Non-identical also it will be there. Sometime what will happen is one of the allele may be modified due to mutation. So this you can see in normal type, homozygous condition, these alleles are there. Heterozygous condition, these alleles are there. In allelic pair, these are identical alleles and this one is non-identical. Sometime what will happen is one of these alleles may be modified. So because due to mutation. Mutation, we know that there is a change in the genotype of organism we are calling as mutation. Like this mutant, okay, for example, caps T is there. Either this caps T become caps T small t or it becomes small t also. So here the caps T small t is there. This caps T small t it may become, because of mutation, become small t also, right? So this dominant allele you can call it as wild type. So the dominant allele and recessive allele only you can call it as mutant type. The dominant allele always changes to due to mutation, it becomes So either heterozygous dominant or maybe recessive homozygous recessive mutant type. Okay. Actually this one you can say it is a wild type or dominant type. This one you can say mutant type. Small t is the mutant type. For example, suppose we will take example one here. One normal allele is there. That normal allele, it produces the enzyme necessary for the transformation of substrate into product. For example, here you can see there is one allele. This is a dominant allele. This dominant allele which produces one normal enzyme. So this normal enzyme, this enzyme may be needed or required to transform substrate into product. Substrate into product. Normally if the gene is expressing. So this enzyme transforms the substrate into product. If the allele is modified because of mutant, the modified or mutated alleles, they are responsible for production of. This is a normal allele producing normal enzyme. That enzyme required or needed for the transformation of substrate into product. Okay. If this normal allele, normal allele either may be homozygous or may be heterozygous condition. If the normal allele is modified, it becomes mutant allele. And mutant allele responsible for production of, first either it produces, the mutant type either produces normal enzyme or less efficient enzyme. Are you clear with this? Less efficient enzyme. So normal enzyme or maybe less efficient enzyme. Second case is either it may be responsible for production of the enzyme non-functional enzyme also. Non-functional enzyme. And third cases, we can say here no enzyme at all. No enzyme. Are you clear with this? Normally there is a one gene pair which is responsible for which if it expresses it produces one enzyme. That enzyme can transform substrate into product. So this enzyme involved in normal growth and development of the organism. So here normal allele is there, it producing normal enzyme, so that transforming. If this allele become modified, so what will happen? The modified allele only, actually this is a normal allele, also you can say unmodified, and modified also you can say mutant allele. The mutant allele, what will happen? Either this allele, either it modified like this, caps A, small a, or small a also. So this modified allele responsible for production of either normal or less efficient. Either it produces normal enzyme or maybe less efficient enzymes also. Or another cases non-functional enzyme also. Another cases no enzyme at all. If you take case 1, this unmodified allele, so this modified allele which is producing enzyme which is equivalent to unmodified alleles. Okay, here producing normal allele. How this normal allele unmodified producing same type of enzyme produced by this mutant allele also. So this mutant allele also the same phenotype on the in case of silent mutation. Okay, so when it produces normal, if in case of silent mutation, the caps A become caps A small a. That time it produces normal or less efficient, right? You take example case 2 and case 3. Here case 2 and case 3. If it is responsible for production of non-functional enzyme or no enzyme at all, what will happen is, so that the allele is modified. So the dominant allele or wild type allele become modified into mutant type. Are you clear with this? For this one example is the gene responsible for plant height. So tall is there. So this genotype it giving so because the genotype responsible for tallness of the plant. How the plant growing tall? For growth of plant it required so plant hormones, the growth hormone. The growth hormones are encoded by or expressed by this particular dominant allele. This dominant allele it produces growth hormone. If hormone produces, what will happen? This hormone only induces the growth or enhances the growth so that the plant will grow tall. If this allele is modified, what will happen? It modified like this, or maybe it modified like it's mutated, small t, then so it produces normal hormone or maybe no hormone or non-functional hormone. So if it is normal hormone, okay, it produces tallness. If it is non-functional hormone and no hormones, definitely, so the hormone is not available, so that what will happen is the amount of hormone may be less or maybe no hormone, so that the plant height become dwarf. Okay, so plant will not grow further, so the plant will be dwarf, right? Are you clear with this concept of dominance? One more time I will explain. So the concept of dominance, all diploid organism, they are having copy of gene, and each copy of gene. So in that pair of gene, you can call it as alleles. The allele may be identical or may be non-identical. So one of them may be modified in that allele. So the allele, if modified, the gene either become mutant allele or modified allele. Normal allele produces normal enzyme that is needed for transformation of substrate into product. But modified allele either produces normal or less efficient or non-functional or maybe no enzyme at all. So when it produces normal less efficient, if the mutation is if it is silent, or if the normal allele modified into mutant type, then it produces only non-functional or no enzyme at all. So no enzyme at all. Okay. So this is the concept of dominance. I hope that you understood, right? And thank you for listening my botany class. And we will see next video.